AP 120, Chapter 14, Topics, Antibodies, Classifications of Immunity, and Immune Disorders. So, let's go into more detail about antibodies. To start with, there are 5 major types of antibodies. Antibodies are also known as immunoglobulins and also known as gamma globulin. So, antibodies, also known as immunoglobulins or gamma globulin. And antibodies tend to have this Y shape that they're associated with, sometimes multiple Ys, sometimes just one. There is the constant region, which is basically the same from antibody to antibody. And then there's the variable ends that alter. It's these variable ends where the antibody can bind to its specific antigen. So, first up, IgG. IgG is normally found in tissues and in the bloodstream. It can bind to bacteria cells, viruses, and toxins. It deactivates toxins and viruses because it's so big relative to the virus or toxin. The virus or toxin can't function properly. It also targets these things for phagocytosis so that macrophages or neutrophils are easier for them to find the viruses or toxins, to engulf them, or to notice the bacteria cells and engulf them. Also, especially for bacterial cells, IgG can stimulate complement in the blood. Again, complement are proteins already found in the blood plasma, and those proteins can do many things, including punching holes into bacterial cells. So again, here is the hole formed by complement proteins that put a hole in the bacterial cell, making it deceased. Complement also can stimulate inflammation and attract other phagocytes to the area. IgA. IgA is in exocrine gland secretions, so it's in breast milk, saliva, tears, nasal fluid, etc. So it has the opportunity to stop pathogens, to bind to their specific pathogen before it even gets into the body. So having, say, you could have 4 different pathogens bound together, forming a larger group that may then be unable to get into the body and cause infection. Also, IgA being found in breast milk can give newborns passive immunity. IgM is found in blood plasma. It can also activate complement. It can also lead to agglutination. So you see it has 1, 2, 3, 4, 5 of the Y-shaped subparts, and each of those could bind to a different antigen. So that's a lot of potential cross-binding leading to agglutination. And it is IgM antibodies that are the ones that bind to the antigens on the surface of red blood cells. So again, if you are blood type B and you're giving a transfusion of blood type A, the anti-A antibody will bind to A, and then that can cause macrophages to arrive, that can cause complement proteins to cascade in, can do a lot of different things that would cause problems. All right, IgD. IgD is on the actual surface of the B lymphocytes. So remember, the B lymphocytes are able to bind antigens, either on a pathogen or on a antigen-presenting cell like a neutrophil or a macrophage. And that surface receptors are actually antibodies, IgD to be specific. IgE is another exocrine gland secretion antibody, so it's found in the secretion of some exocrine glands. And one thing it can do is promote the allergic reactions in the body. So normally when it functions against an actual pathogen, that can be useful, but when it's targeting an allergen, that is a waste of time because allergens won't hurt us. We hurt ourselves instead with our allergies. And in the process, the IgE can stimulate mast cells that produce a lot of chemicals like histamine that cause the allergic symptoms. All right, so again, antibodies can cause agglutination. They can bind to multiple antigens, pulling, connecting, grouping together, say, bacteria or, for instance, blood cells, things like that. Activate complement. Again, complement is able to attract phagocytes, stimulate inflammation, and cause lysis of pathogens. Activation of complement also leads to opsonization. Opsonization just means that it makes the pathogen easier for the phagocytic cells to find, so easier for macrophages and neutrophils to find and destroy. Initiate inflammation. So antibodies can also help in initiating inflammation. And this, of course, causes the changes in that area that we would expect: pain, swelling, heat, redness, more white blood cells, leukocytes being attracted, potentially isolation of the infection site, all that sort of stuff we talked about in a previous video. So the first time you are infected, you have your first exposure, then you eventually have your primary response, and if all goes well, you will eventually defeat the pathogen. Unfortunately, primary immune responses often don't prevent us from getting sick. So we still become ill or sick because of the infection. However, we do form memory cells that will remember the infection for future times. Then when we do have a future infection, the memory cells can cause a more rapid and long-lasting response. So you get a much, much stronger response against the presence of that antigen. And then it ramps down much, much slower. Often, this will allow us to not have any symptoms. So we get infected with the pathogen, but we do not suffer any illness. We do not suffer any symptoms. And that is what we call immunity. All right, types of immunity. There's naturally acquired active immunity. That's what we've been talking about. That's where you have the normal response to the initial infection by a specific pathogen. We will get sick, and then we will have memory cells so that if we are exposed to that pathogen again, we will be able to respond faster. So this is normal. This is how we normally get immunity, is by getting sick. There's artificially acquired active immunity. Artificial acquired active immunity is when we use vaccines to mimic exposure to the pathogen. This allows us to mount that immune response, to develop those memory cells, but we in the process get to avoid actually getting sick. So the vaccine has the antigen in it, goes into the bloodstream, stimulates an immune response, and including the production of memory cells that will hopefully activate and protect us from future infections by that pathogen. Then we have artificially acquired passive immunity. Artificially acquired passive immunity is when we have basically antibodies from something else injected into us that will fight the pathogen. So this is, say, an anti-serum. We know some of the treatments for COVID has been to have people who were infected and recovered have their blood plasma isolated and then injected into people who are severely ill to help fight the pathogen. It's artificial because we have to have these antibodies come from something else from that serum. It's passive because it's short-lived. There are no memory cells stimulated. It won't help protect us in the future. Examples of when this is used includes snake bites and tetanus. And then there's naturally acquired passive immunity. This is specific to when the mother passes on her antibodies, her IgA antibodies, through breast milk to the infant. Again, this is short-lived, only lasts as long as the infant is breastfeeding, the time period in the infant's life in which it is breastfeeding. And so it's a short-lived and doesn't stimulate any immune response cells. So there's no memory cells that are going to be involved. Allergies. Allergies are when our body has an immune response to something called an allergen that is not bad for us, that is not going to cause problems. So we have an excessive immune response to something that's not dangerous. It can include, say, the venom in bee stings, dust, pollen, pets, furry animals, some foods, nuts, gluten, grass, etc. Now, there are different ways you can have an allergic reaction. There's the immediate reaction allergy. This is where you have an inherent ability to overproduce IgE. IgE then causes mast cells to be activated and they release chemicals such as histamine that cause the symptoms of an allergic response, such as vasodilation, so you get this rash, swelling of that rash, in the respiratory tract, the contraction of bronchioles, increased mucus production, and so on. So this is an immediate response to exposure to that allergen. And this can sometimes be extremely severe and can cause the person's body to be flooded by those allergy mediators, those chemicals like histamine. And this can cause something called anaphylactic shock, which can even lead to death. And this is why, say, people allergic to bee stings or have severe food allergies will carry EpiPens with them. They can inject the adrenaline in to try to counter the effects of the allergy mediators. Immediate reaction allergy. There's also the delayed reaction allergy. This one requires repeated exposures before you start having inflammatory reactions. And it can take time. So you might be exposed and then 24 to 48 hours later, that's when you actually start to have the allergy response. And often a lot of the allergens that are tested for in the allergy test, where they inject tiny bits of different allergens into your skin, often these are delayed reaction allergy responses. So you get the injections and have to wait a while before you see the results. Treating allergies. Well, there's avoiding the allergens, treating the symptoms, antihistamines, decongestants, gestants, epinephrine. There are some mast cell inhibitors medications out there to suppress the mast cells. And there are allergy shots. Some allergies, it turns out you can try to build up a resistance to those allergens by injecting or eating small amounts of them over time. Your body will start giving you IgG antibodies against the allergens, and this will help to bind up allergens before the mast cells and IgE antibodies can respond. Transplantation and tissue rejection. Well, if you don't have a good match between the donor and the person receiving the recipient, then you end up having a tissue rejection reaction where your own immune system destroys the transplanted tissue or organ. So you need a close match between the donor and recipient, specifically the MHC antigens. Those are the ones you really want to make sure match really well. Also, transplant recipients also receive immunosuppressant drugs that suppress their immune response to try to decrease the likelihood of transplant rejection, although it does make them more sensitive, more likely to suffer from infections from other pathogens. Autoimmunity disorders are disorders where our immune response attacks our own tissues and cells, where we're producing antibodies against our own antigens. This is not good. And then we activate cytotoxic T cells that will start destroying our own cells. Again, very, very not good. They may be stimulated and evolve from a viral infection where the virus had antigens similar to your own. Could be from faulty T cell development or some other non-self antigen that's similar to the self antigen. Some examples of autoimmune disorders include lupus, type 1 diabetes, rheumatoid arthritis, psoriasis, Crohn's disease, and so on. That is it for chapter 14.